Hi, this is Dr. Rosario. In our last video lecture, we talked about some of the general principles behind microbial control practices. In this video, we are going to focus specifically on physical methods of microbial control. And then in our next video, we will talk about chemicals that we use to control microorganisms. So in this video, we're going to talk about a variety of different physical methods that we use to control microbial growth. We're going to start out with heat and then cold, desiccation, hypertonicity, lyophilization, ionizing radiation, UV radiation, and then finally filtration. These different methods each have advantages and disadvantages when it comes to controlling microorganisms. And we're going to talk about each one, kind of define what they are and some of the common applications that we see. Heat is one of the oldest and most common physical methods of microbial control. In our module on microbial growth, we discussed the role of temperature in promoting microbial growth. Each microorganism has an optimal growth temperature, which is largely dependent on the function of that organism's metabolic enzymes and membrane stability. Heating organisms to a temperature above their optimal growth temperature can lead to protein denaturation and alteration of the cell membrane. In general, these effects are microbiocidal. They're going to kill the microorganisms. There are two important metrics that can be used to characterize an organism's heat susceptibility. The thermal death point is the lowest temperature at which all cells in a population are killed following a 10-minute exposure. This is helpful when comparing the sensitivity of different organisms to heat. For example, the thermal death point of endospores is much higher than that for vegetative cells. The thermal death time is the length of time needed to kill all cells in a population at a specific temperature. This is useful when trying to decide which heating method is appropriate for your particular situation. Heat is a form of energy. Moist heat is transferred from hot water or steam. Dry heat is transferred from the air. Water is more efficient at transferring this energy than air is. To underscore this point, the thermal death time for moist heat at 121 degrees Celsius is 15 minutes. At the same temperature, the dry heat takes 600 minutes to sterilize a specimen. Moist heat results in denaturation and coagulation of proteins. Dry heat, on the other hand, dehydrates cells at lower temperatures and oxidizes biological molecules to ash at higher temperatures. The three most common forms of moist heat used to control microbial growth are boiling water, pasteurization, and autoclaves. Boiling water is one of the oldest and easiest methods of eliminating microorganisms. Boiling does not require any specialized equipment other than a heat source like an open flame or a stovetop. After hurricanes or natural disasters, we often hear the health department advising people to boil their water to ensure that it is safe to drink. Medical instruments and even baby bottles can be disinfected with boiling water. Boiling water, it is important to remember, does not completely sterilize. At sea level, water boils at 100 degrees Celsius. This temperature is high enough to kill most vegetative bacteria, viruses, and protozoans, but not usually endospores or protozoan cysts. It is important to remember that water boils at lower temperatures at higher altitudes. These temperatures may be low enough, particularly up in the mountains, like in the Rocky Mountains or out in Colorado. These temperatures may be low enough that protozoan cysts can survive. And this has been linked to cases of Giardia lamblia or our favorite, beaver fever, where hikers have been out in the woods, they've boiled their water, they think it is safe, but it's not because they're not eliminating the cysts in that situation. So remember, water boils at 100 degrees Celsius at sea level, but at higher altitudes, the temperature that it boils at is lower. Pasteurization is named for the person that invented the process, Louis Pasteur. He developed pasteurization to prevent spoilage of beer and wine, which are sensitive to heating. Today, pasteurization is used to kill heat-sensitive organisms in a variety of food products, including milk and juice, without negatively affecting the flavor or the texture of the foods. The process involves rapidly heating and then cooling liquids over a series of several cycles. There are many different pasteurization methods with different levels of effectiveness. And it is important to keep in mind that pasteurization does not sterilize substances. Okay, that's real important. Think about that. You buy pasteurized milk at the grocery store. So pretty much all of the milk that we buy off the shelf at the grocery store has been pasteurized. But even if you don't open that milk and you keep it in your refrigerator, it's eventually going to spoil. That spoilage of your milk is due to bacteria that are growing there in the milk. So the milk's been pasteurized, but it has not been sterilized. Okay, pasteurization is a way to limit the number of microorganisms present in a substance without compromising taste or flavor. So you can see in this picture, there's a series of coils in this machine, and that's part of the rapid heating and cooling process that occurs through pasteurization. An autoclave provides moist heat under pressure. Under atmospheric pressure, steam is 100 degrees Celsius. That's the boiling point of water. In an autoclave, that steam is pumped into an enclosed chamber that allows for the pressure to build up, and that raises the effective temperature of the steam. Most autoclaves have a standard operating temperature of 121 degrees Celsius, which is high enough to inactivate endospores and sterilize materials. Autoclaves are commonly used to sterilize laboratory equipment, culture media, medical equipment, and other materials that can withstand the high temperature and pressure. So think back now to what we've already talked about. So remember I said moist heat at 121 degrees Celsius takes about 15 minutes to sterilize materials, but dry heat at that same temperature takes 600 minutes, right? So that's not practical. Our autoclave can sterilize materials in a relatively short amount of time. Think also about boiling and pasteurization. Neither one of those can effectively sterilize your materials. They can disinfect, they can reduce the numbers of microorganisms, but they cannot sterilize. But autoclaving can. Few materials can withstand dry heat sterilization without being destroyed. Hot air ovens are sometimes used to sterilize equipment instead of an autoclave. These ovens, however, require higher temperatures and longer periods of time because air is less effective than water at heat transfer. Incineration oxidizes biological materials and cells. We use incineration in the laboratory when we flame our loops. Incineration is also commonly used to destroy medical waste. Unfortunately, this can also create a fair amount of air pollution. So we've talked about heat. Now let's go the opposite direction and talk about cold. Something we all have in our homes is a refrigerator. Treatment slows down microbial growth. It is generally bacteriostatic. Likewise, freezing further slows down microbial growth. We use our refrigerators and freezers to prevent the growth of pathogens and spoilage microorganisms and preserve our food. It is really important to take into account how long it takes for food items to cool down in the refrigerator. Hot foods should be divided into small, shallow containers before being stored in the refrigerator to allow for the most rapid cooling. If you put a large pot of chili directly in the fridge, it might still be warm in the middle by the next morning. This can be problematic because pathogens can grow in food that has not cooled properly. So it's important to divide those materials up as we're applying our cold treatment. So that's one of our limitations of this method. Another important limitation, of course, is that we have to make sure that our refrigerators are working properly. If they're not working properly and they warm up even just a little bit, that can be enough to be permissive for microbial growth. The process of drying foods and adding salt have been used to preserve foods throughout human history. Removing water removes the water from the cells, and that inhibits metabolism. By doing so, we inhibit microbial growth. So, that classic beef jerky has no water in it, and that meat, which normally could rot if it was fresh meat, will remain edible for an indefinite period of time because bacteria simply cannot grow there. Lyophilization is another interesting way of preserving bacteria in particular. So lyophilization is a fancy way of saying freeze drying. Lyophilization is a method that we use to preserve bacterial cultures. Again, it's a form of cold application. You've got the freeze part, but you also have the drying. So you're freezing and you're removing water from your substances. And we can use this to preserve bacteria almost indefinitely in the laboratory. So if you have a strain of bacteria that you're studying and you want to make sure that you have it for the future, you can lyophilize it. And that's what you're seeing in this picture is a series of vials of lyophilized cultures. Okay, so let's move away from heat and cold and talk about radiation. Radiation consists of energy that has both an electric and a magnetic field associated with it. Different kinds of radiation are associated with varying wavelengths and levels of energy. So we are most familiar with visible light, which is kind of right in the middle of this figure. But there are lots of other forms of radiation and several that are useful in controlling microbial growth. Ionizing radiation and ultraviolet radiation both have shorter wavelengths and higher energy and are capable of damaging cells. So those are the ones that we're going to focus on here. Think back to your basic chemistry lessons. An ion is an atom that has a charge associated with it. X-rays and gamma rays are forms of ionization. They have the most amount of energy associated with them, the shortest wavelengths, and they can cause atoms to lose electrons and become ions. Ionizing radiation can easily penetrate through many materials like plastic, paper, water, even the human body. Right? Because of this, X-rays and gamma rays are both used in medicine for imaging. As ionizing radiation passes through cells, it can damage important molecules like DNA and proteins. Double-stranded DNA breaks caused by ionizing radiation can lead to cell death or to permanent mutations. This is why we have to limit the amount of our exposure to this form of radiation. Ultraviolet radiation, on the other hand, has longer wavelengths and is not able to penetrate solid barriers like paper, plastic, and living tissue, etc. Ultraviolet radiation, or what we consider ultraviolet light, is able to interact with cells at a surface level, and it can damage cells by causing what's called a thymidine dimer to form, or a thymine dimer. So in the DNA, if you have two thymines next to one another, they can actually form a covalent bond between them as a result of exposure to ultraviolet radiation. This damage, if there's just a little bit of it, can be repaired by the cell's DNA repair enzymes, but if there's too much ultraviolet radiation, that can permanently damage the DNA. It can ultimately lead to cell death, or in the case of our own cells in mammals, we can have damage to our DNA that results in skin cancer, right? So ultraviolet radiation, whether you're talking about a bacterial cell or a human cell, can result in thymine dimers that can damage DNA. So just like ionizing radiation can damage DNA and proteins in cells, ultraviolet radiation can also damage cells, specifically by damaging the DNA. The big difference between ionizing radiation and ultraviolet radiation is that ionizing radiation can penetrate through surfaces, where ultraviolet radiation can only penetrate very shallowly into surfaces. So really can't go very deep at all. It's not able to get past barriers like plastic. Filtration is a disinfectant technique that does not directly destroy microorganisms, but instead it physically removes them, right? So it's the direct removal of microorganisms from a substance. We can use filtration to filter air or to filter liquids. HEPA filters are used to filter out airborne microorganisms. You can find HEPA filters in a variety of different applications, from air conditioners to vacuum cleaners. These are high efficiency particulate filters. And then membrane filtration, on the other hand, can be used to remove microorganisms from heat-sensitive liquids. Some liquids can't be heated without being destroyed, so we can't autoclave those liquids, but instead we can filter them. And filtration allows for us to remove microorganisms without affecting the quality of that particular liquid. Okay, so we've talked about a lot of different types of physical methods of control. I have a couple of images for you. So this is what a HEPA filter looks like. It's a very complex structure that allows for air to flow through, but doesn't allow for small particulates to flow through. So usually we're talking about substances all the way down to 0.1 micron in size. So remember your bacterial size, right? Most bacteria are about 2 microns in size. So a HEPA filter will prevent most bacteria from getting through, and sometimes even viruses. Finally, this is examples of our membrane filters that can be used to filter out our liquids. The one that you're seeing with the pump next to it can be used to filter larger amounts of liquids, and then you have a syringe filter on the other side here, where you can filter out 10 milliliters of a fluid at a time. Okay, so in our next video we will talk about our chemical methods of control.